Hey Mustangs, in this video we're going to take a look at the cell cycle. So we're going to be looking at the life of a cell basically. We're going to look at how a cell is created, we're going to look at what happens during the life of that cell, and we're also going to take a look at what happens when a cell is going to divide to create more cells. So before we actually get into it, I'm going to show you a short video clip that actually shows some cells dividing and kind of the background history of how we learned about the cell cycle.
Our next great discovery sought to answer a question at the heart of biology: how cells form new life. You know, in the course of human events, this is all pretty recent, right?
Yes.
We found nuclei, there's cells around nuclei, animals and plants have cells.
Yes.
Then what happens next?
Well, after a gap of some 30 years or so, people began to see cells dividing.
Cell division is the process that the cell of a plant or an animal goes through to duplicate itself. While scientists had known about the process for decades, German zoologist Walter Flemming was the first to describe it and to publish his findings.
And his book was published in 1881. This book was momentous and extremely important in the history of cell biology because Flemming described cell division in a way which would be acceptable in a modern journal.
Flemming observed the process of cell division in great detail, with the help of a powerful new microscope and new dye staining techniques. With these innovations, he was able to identify structures that later were named chromosomes. And then, his great discovery. During cell division, the chromosomes undergo dynamic change. They divide into two identical parts, one for each daughter cell. Flemming called this process mitosis.
So that's the whole process of mitosis as he described it in 1881.
In a tadpole tail.
In a tadpole tail, yes.
And he knew to go to the tail because the tail is growing, or the back end of a tadpole, there's a lot going on.
Exactly, yes. He knew that if you're going to see cell division in detail, sorry for the pun.
I like the pun. Kooky for the pun.
Then that was one of the places to look.
The splitting of chromosomes during mitosis was a breakthrough discovery. It opened the door for scientists to begin understanding how one cell can turn into a complex organism made of many interacting cells.
Alright, so before we get into the full cell cycle here, I want you to think of why would cells need to divide. Okay, now there's two main reasons as to why cells would need to divide. So I'm going to give you just a moment here. Think about it. So what could be two big reasons why cells would need to divide in your body? Alright, I know that was a little bit of a short minute there, but let's go ahead and take a look. So the first reason is cells divide to cause growth. So in order to grow, you must add new cells onto your body. So for every single living thing on our planet that is a multicellular organism, we all started off as a zygote, when the sperm and the egg came together and formed our very first cell, and that cell divided, and then those divided, and we began to grow. So growth is adding cells onto the body. So for us, a human being, going from a child, a baby, all the way up to adulthood, in order to get bigger, in order to grow, we have to have our cells divide to add more cells onto our body. If that doesn't happen, then we do not grow. We have to add more cells onto our body. The second reason for cell division is to replace dead or damaged cells. So all the time, cells in our body are dying. And if we didn't replace them, instead of growing, we'd get smaller and smaller all the time if we didn't replace those dead cells. Another reason, damaged cells. Okay, so our cells get damaged all the time. If you've ever gotten a cut or a scrape, you've destroyed cells. And in order to replace those cells that have been damaged, you have to go through cell division. The cells that have not been destroyed divide in order to fill in a gap such as this. right, so the cell cycle is a real simple series of predictable steps. So we can actually divide it kind of into 4 sections here, but there's actually 2 main parts. The first 3 parts of the cell cycle are basically when a cell is not dividing. And then we have 1 section here that's going to represent when the cell actually divides in the life of a cell. Okay, so on your paper, I'd like you to label each section. The first section, go ahead, label G1. The second section over here, label S. The third section, label here G2. And this last section, label M. Okay. Now, before we discuss this in the video, I'd like you to please take some time right now and turn to page 155 in your biology notebook and read the section, the cell cycle and interface. Okay, so those 2 sections on page 155. So go ahead, pause the video, read that, and then come back. All right, so here's what you should have gotten on your paper. Cell grows bigger during G1. Okay, so after a new cell is formed, it's going to grow bigger to reach its mature size. A cell will make a copy of its DNA during S phase. So this is S phase here. And then here, during G2, the cell makes final preparations to divide. So these first 3 phases here are called interphase. So on your paper, if you could put on this arrow right here, so just write it right onto this arrow, write interphase, I-N-T-E-R-P-H-A-S-E. So that's the first 3 phases right here. So that's where cells spend most of their lifetime, during in these 3 phases here. This section here, this is when a cell is going to divide. So this is cell division. Okay, so the cell prepares to divide here. It made a copy of its DNA here, and it actually grew to maturity here. So going from here back the other way, cell grows bigger to maturity, makes a copy of its DNA. Now what we're talking about here is making a copy of the chromosomes. Now that we know what chromosomes are, they're just DNA wrapped around histone proteins. So it makes a copy of all of the chromosomes, makes final preparations to divide, and then here the cell will finally divide. Now, since we're going to be looking at cell division next class, what actually happens to the chromosomes during cell division, during mitosis to be specific, and meiosis, we need to understand what's going on with the chromosomes during cell division. So here, remember, this line would represent 1 single chromosome. And remember what a chromosome is. So it's a whole bunch of DNA, specific instructions for making specific proteins right here, wrapped around histone proteins. Now, when we make a copy of 1 chromosome, it's going to look like this. So if I make a copy of this entire chromosome, of all the DNA instructions here, it's going to look like this. This half here is an exact copy of this half here. But the copies stay stuck together for a short period of time. They're not going to stay stuck together forever. What we want to happen is that when the cell finally divides into 2 cells, that we break these copies apart. This copy goes into 1 cell. This copy goes in the other cell. Now both cells have the exact same DNA instructions, a copy of each chromosome. Okay, so a few new vocabulary words here. So each half of a copied chromosome, so like I said, this is an exact copy of this half here. We call them sister chromatids. Okay, so that's a sister chromatid and that's a sister chromatid because they are exact copies of each other. So the definition of sister chromatid is an exact copy of the DNA. So sister chromatids are exact copies of each other. Another word, centromere. The part where the copies stay stuck together, that is called the centromere. Okay, so the centromere, the spot where 2 sister chromatids hold together. Okay, what I'd like you to do right now is real quickly, you're going to pause the video in just a moment and write these down on your chromosome chart, vocabulary chart. So on that chart already you should have homologous chromosomes, the sex chromosomes, and autosomes. Okay, so go ahead, pause it for a moment and write down those 2 definitions. All right, now to help you a little bit with a little bit more of a visual aid here, here's a picture of Megan Fox. So here, if you wanted to make a copy of Megan Fox and she was a chromosome, it would look like this. The 2 copies would stay stuck together. Okay, so here's 1 sister chromatid, that's 1 copy. here's the second sister chromatid, the exact copy, and they're stuck together right here at the centromere. Kind of like Siamese twins, I guess you could kind of think of. So they'd be like Siamese twins stuck together. But during cell division, we'd want to break apart those twins so that way one copy goes into one cell and one copy goes into the other. Alright, so now let's take a look at what it looks like when homologous pairs get copied. So here we have a homologous pair. They are the same size, same shape, and they have genes in the same place. Now, a lot of people want to say, okay, if they're the same size, same shape, with genes in the same place, they must be identical. Well, they're not. They're very, very similar to each other. Okay, so that's why I picked two actresses here. Now, this one here, this chromosome here, represents the chromosome of the homologous pair that came from dad, and this chromosome here represents the chromosome in the homologous pair that came from mom. Okay, because in a homologous pair, same size, same shape, genes in the same place, one always comes from mom, one always comes from dad. Okay, now when we make a copy of these homologous pairs, here's what it looks like. Okay, so we make a copy of the one from dad, and the copies stay stuck together. So here's one copy, and here's the exact copy right next to it. And we remember from the last slide, this is called a sister chromatid, and that's a sister chromatid. Same thing happens with the chromosome from mom in the homologous pair. So we make a copy, and the two copies stay stuck together. So there's a sister chromatid there, there's a sister chromatid there, and they're held together right there at the centromere. So we've made a copy of both within the homologous pair, the one from dad and the one from mom. So how are sister chromatids different from homologous pairs? So I want you to think about that and write your answer on your paper below the box where you drew this, and go ahead and we will discuss this in class when I see you next. Alright, so quick reminder of the cell cycle, what we've talked about so far. Cell grows bigger during G1. During S, this is where we make a copy of the chromosomes, a copy of all the DNA. And G2 is when the cell divides, and then here, this is where the cell actually will divide. So during S phase is when we make copies of the chromosomes. So the last two slides we looked at how does it look when we make a copy of one chromosome, and you get the sister chromatids stuck together like that in the middle. So one sister chromatid, the other, and they're stuck together like this. Okay. And then you have when a homologous pair gets copied, same thing, they stay stuck together, the two copies. Okay, so if we look at a cell that has homologous pairs in it, I want you to think about the answer to this real quick. If we have a cell that has homologous pairs, is it diploid or haploid? Okay, diploid. So if we make a copy of all the chromosomes, because the cell is going to get ready to divide, so if S phase occurs, remember S phase when we make a copy of all the chromosomes, the cell will now look like this. Okay, so we made a copy of every single chromosome, and the copies are still stuck to each other. Okay. Here's another example. So here's a cell. It's a diploid cell because there's homologous pairs, and we're going to say S phase occurs. And I want you to draw a picture of this on your paper of how it would look. So go ahead, take a little bit of time, pause it if you need to, and draw the picture. And you should have gotten this. So this is how this cell would look after S phase, not during S phase. So this is how that cell would look after S phase. We made a copy of every single chromosome, and the copies are still attached to each other. And last bit of review here, those copies are called sister chromatids. So this is a sister chromatid, and that's a sister chromatid for this chromosome here. Here's a sister chromatid and a sister chromatid here for this chromosome. Sister chromatid of this chromosome. Sister chromatid of this chromosome, and those sister chromatids, those copies, are held together right there at the centromere.